Okay, hi guys. Clearly, I'm not Dr. Pochynyuk. My name is Natme. I'm a postdoc in Dr. Pochynyuk's lab. I'm here to tell you a little bit about our lab and why it's a great place for any ambitious graduate student, whether you want to join full-time or you just want to do a rotation with us. I remember back when I first joined the lab as a postdoc, my first thought was that I wish I picked this lab for my PhD years. And the main reason is we publish a lot of paper every year. I don't need to tell you how important it is to have a great publication record as a graduate student. Just this past year, we submitted 6 papers. Most of them is already published, and the last 2 are under review in great journals. So that's a really good plus for any students that want to join. So a little bit about our research interests. Our main research focus is about renal physiology. We study ion channels in the context of several kidney diseases like hypertension, hyperkalemia, polycystic kidney disease. The method that we use to study them is called patch clamp technique, which is one of the only single molecule methods that it's out there, which allows you to study a single molecule, a protein, in our case, a channel. You attach an electrode to the cell membrane and you can tell whether a channel is open or closed. You can apply drugs like agonists and inhibitors and study the function of the channel in response to them. And this is very helpful because a lot of these channels are implicated in a variety of kidney diseases. So this would give us an edge in coming up with different therapeutic approaches later on. We do other techniques in the lab. We routinely do Western blot and a lot of fluorescence microscopy, which you can see an example of them that we have done in the slide below. We do a lot of cell culture as well. We transfect different cell lines like CHO cells or more kidney-specific cell lines, and we overexpress a protein in them, use them for Western blot or imaging later on. However, the majority of our work is done on animal models, mostly mouse and rat, whether it's a knockout or wild-type model. In fact, right now we are in the process of creating a double knockout, which is a good opportunity for anybody who wants to learn about genotyping and PCR, which is useful methods later on for your career. About our lab environment, we are very friendly and fun. We love to help each other out in solving the problems that we approach in our everyday techniques that we use. There is no toxic competitive behavior whatsoever, and everybody collaborates with each other on several projects. So if you join, you have the chance to work on multiple projects, as well as learn from several postdocs that we have in the lab that gives you a lot of experience for later on. And that's pretty much it. I'm here to answer any questions you have. Hopefully this was a good summary about our lab, and you can contact Dr. Pochynyuk with further inquiries that you have.